This is a molecular machine that cuts DNA. Evolution spent something like 1 billion years shaping it, one small mutation at a time. Then, a computer was allowed to rewrite much of it in a single design run. It kept a small set of load-bearing parts fixed and redrew everything else from scratch. And some of those redrawn versions did something the original could not. They edited human DNA better than nature's own version. Real cells, real cuts, measured in a dish. So, straight answer to the question you are already asking. Did an AI just design a better gene editor than the one evolution built? In the narrow way this paper tested it, yes. But keep the caveat, because it carries the whole story. Better means it cut its intended targets more often, in bacteria and in human cells growing in a dish. It does not mean a finished therapy, and it does not mean safer. The genuinely surprising part is how they pulled it off. The work comes from Jennifer Doudna's lab at the Innovative Genomics Institute at UC Berkeley, led by Peter Skopintsev, and it was published in Science in July 2026. The enzyme they started with is called TnpB. In plain terms, it is a tiny protein that carries a short strip of RNA, like a search string, uses it to find a matching sequence in a genome, and cuts the DNA right there. That is the same basic trick CRISPR uses, and TnpB is thought to be an ancestor of the Cas12 editors, a stripped-down version of the machine from before CRISPR immune systems even existed. Stripped down is exactly the appeal. The Cas9 protein most people have heard of is bulky, around 1,300 amino acids. TnpB does a similar job with about 400. And size is the quiet bottleneck in gene therapy. The editor has to be delivered into cells, often packed inside a virus shell with a strict cargo limit. A smaller editor leaves room for everything else you have to send in with it. Here is the design tool at the center of it. You have probably heard of AlphaFold, which takes a protein's sequence of amino acids and predicts the 3D shape it will fold into. The models used here run that in reverse. You hand them a shape, the backbone of the folded protein, and they hand back sequences that should fold into that same shape. It is called inverse folding. The shape stays fixed. The sequence is free to change. But a single shape has a huge number of sequences that could fit it, and most of them would break the enzyme. A protein has a few load-bearing residues, the exact atoms that carry out the chemistry, or that grip the RNA and the DNA, and if the model touches those, the machine dies. So the team did something clever with evolution. They lined up thousands of natural TnpB relatives and asked which positions almost never change across all of them. Those positions became a locked list. The cutting core, 3 specific acidic residues that actually perform the cut, and the spots that hold the guide RNA and the target DNA. Everything on that list was frozen. Everywhere else, the model was told to redraw freely, generating 50,000 candidate sequences at a time. This is the part that sets it apart from earlier AI-designed editors. In prior language model Cas9 editors, such as OpenCRISPR, the short DNA motif binding domains stayed more than 99% identical to the closest natural versions. These designs drifted far past that. In the regions that touch the RNA and the DNA, the best synthetic versions dropped to between 72 and 83% identical to any known natural sequence. In the parts that do the real work, they are genuinely new proteins, not lightly edited copies of what nature already had. Designing sequences is the cheap part. As one of the lead authors put it, a computer can propose millions of designs. The real bottleneck is testing them. So they built nearly 2,000 of these synthetic enzymes for real and ran them through a survival test in bacteria, rigged so that only a working cutter lets the cell live. 466 of them cut. A around 8% of the whole library beat the natural enzyme in that screen. This is AI-guided engineering with a wet lab bolted on, not a program that prints a perfect enzyme on the first try. Then they moved the best variants into human cells. They aimed them at a gene that makes cells glow blue, so a successful edit literally switches the glow off, and they counted how many cells went dark. The natural TnpB knocked out that target in about 28% of cells. The two best synthetic versions reached roughly 46 and 50%. And at some other spots in the genome, the improvement was close to fourfold.
So why would a redrawn enzyme work better than the one evolution tuned? To find out, the team froze a synthetic version mid-action and imaged it with cryo-electron microscopy, the first experimental structures ever solved for an AI-designed RNA-guided nuclease. The images showed the redesigned surface making brand new contacts with the RNA and DNA that the natural enzyme simply does not make, extra points of grip that seem to help it hold on and cut. They even caught a fleeting moment, right as the enzyme locks on to its DNA target, that scientists had predicted for years, but never actually seen. Now, the honest limits, because they are the whole difference between a real result and a press release. Everything here is on-target editing, measured in bacteria, in plant cells, and in human cells growing in a dish. The team did check for off-target cutting, and the best designs looked about as clean as the original, but specificity is not fully solved across every guide and every cell type. Nothing here was tested in a living animal. Getting an editor into a body, immune reactions to a partly synthetic protein, long-term safety, all of that is still ahead. And it still took a large physical screen to find the good ones. The AI narrowed the search. It did not replace the lab. What they have really shown is a recipe. Take a compact molecular machine. Use evolution to mark the few parts you must not touch. Let a generative model reinvent everything else. Then screen hard for the versions that actually work. If that recipe holds for other editors, it points toward gene editing tools built to order, small enough to deliver and tuned for the job, instead of borrowed whole from whatever nature happened to leave lying around. The enzyme evolution had spent ages perfecting turned out to have a lot of slack left in it. The open question now is how much of the rest of the toolkit does too.